Okay, guys, so what we're going to do is work on the assignment that we started on Monday, DNA template for protein transcription. And you're going to be doing this today with Miss Sanders. Okay, so she is going to pass out your work that you did yesterday, the worksheets that you did yesterday. And as we go through these instructions, I want you guys to pause them. I'll give you instructions about a part to start and kind of guide you through that. And then I want you to pause the video while you complete that step. And then when everybody's finished, you can restart the video and move on to the next step. Okay? So first thing, if you guys will all get into your groups that you worked in yesterday and get your work from yesterday that you started on, the copy paper that you started your work on. Okay, so the next thing that we need to do, you should already have your DNA template strand written out on your paper, like I've shown you here. Okay, so I just did the first 4 codons there. You go underneath there and do your messenger RNA transcription. So I'm going to do the first few for you just to make sure that you're, that you're all on the same page. So with an A, you would have paired U, because it's RNA, UGC, okay, and then it's going to be ACG, AUG, AAC, okay, and just keep doing that until you reach the end of your strand. And make sure that you label it as 5' to 3', because remember it's anti-parallel, okay, so that you're creating it in that way. All right, so pause the video and go ahead and finish all of your transcription. Okay, so what I want you to do now is I'm going to pair you guys up together. Those of you that had the same paper, same mutation type, for example, if you were a group B, I'm going to let you pair up with some other similar groups so that you can go over your messenger RNA strand and make sure that your messenger RNA strands are the same. Okay, so that's going to be Justin's group and Jana's group. You are going to work together. Nathan's group and Molly's group, you guys are going to work together. All right, Victoria, Shallon, Abigail, all of you are going to work together. I know that's going to make you have kind of a big group there, but if you all will work together. And then I need Tommy's group, Brooks, and Hannah's. If all of you will work together, and that's is Brooke, Brittany, and Alana. So I'm not sure if it's register or Enlow, but if you guys will work together, you should all have E. Stone's group. You are the only ones that had a D. Lucky you, okay? So you are going to be working, just continue working with your group. You may want to go back through and just double check yourself and make sure that you transcribed the messenger RNA correctly. Okay, so your job right now is to get together as groups, those groups, and you should all have the same paper labeled as B, C, D, or E. And I want you to compare your messenger RNA strand to make sure that you all transcribe them correctly. All right, after you have finished with the messenger RNA strand and made sure, double checked with each other that everything is correct, I want you to look at your codon chart that we have used to do the amino acid bingo and those kinds of things. We've looked at it several times. And I want you to find what would be the start codon. Okay? Find what would be the start codon. That is going to be where you start your protein, start making your protein. So I want you to ignore everything before the start codon. So you can just draw like a single line through that, like I'm doing here, okay? So you can just draw a single line through anything that comes before the start codon. All right, and you're looking on your messenger RNA strand. This is the strand that you're going to be looking at here when you're trying to identify your start codon on your chart. That is where you're going to start making your protein, okay? So wherever you find your start codon, I want you to come beneath there, and I want you to start translating it, okay? So you are going to perform, you're going to create your protein, and that process is called translation. Okay, so find wherever your start codon is, based again on your codon chart, and you're going to come underneath there and write start or whatever the name of that amino acid is. And then you're going to do that for every codon after that. Okay? So, for example, AAC, you would just come underneath there on your paper, and you can do an abbreviation for that particular codon. And you're going to do that for every codon until you get to the stop codon. At that point, when it codes for stop, you will simply write stop underneath that codon and stop right there. You don't have to finish writing the codons for the rest of the DNA strand or the messenger RNA strand. You simply just stop right there. Okay, so I want in your groups, I want you guys to continue working on that. You're going to just keep working together to look at your codon chart. You can do them individually on your own paper, and you do need to do that. All the original groups need to write down whatever those amino acids are underneath your codons, okay, using your codon chart, but then go back and compare to each other to make sure that you didn't mess up, okay? So you're going to find the start codon, start there, and then find the stop codon, and that is where you're going to stop translating. Okay, now it's time to build our protein, okay? Rather than having each small group make a protein, I'm going to let you guys build your protein all together, okay? So it'll just be one protein built for B. It'll be one protein built for group C, one protein built for group D, one protein built for group E. Okay, so if you'll look at the bottom of your instruction sheet, it'll give you some shapes and codes for those various amino acids. So like, for example, earlier we had done AAC was asparagine. So if you'll look on your paper and find asparagine, okay, on your instruction paper, it tells you that the abbreviation is ASN and that it is represented by a white triangle. Okay, so what you're going to do is you're going to go get a white triangle, and you're going to tape that to whatever is the next amino acid, okay? And on that shape that you get, all right? So let's say that one says it's a white triangle, okay? So on that white triangle, you would put ASN, okay? And I want you to tell me at the bottom of that also what your codon was. For example, AAC was the codon that would be on that one, okay? If you had, for example, let's see, CGC, okay, is represented by a white square, okay? So if you had that codon, it's arginine, and that's represented by a white square. Actually, I did that backwards. Your CGC will be down here. Your codon will be there. And your arginine abbreviation will be there, okay? It's late, guys. Okay, all right, so anyway, so that's the way I want you to do that. You're going to put your amino acids in order, just like those transfer RNAs would be bringing in those amino acids. You're going to put those amino acids in order and build your protein. You're going to tape them together, tape the shapes together, okay? And in taping them together, those represent your peptide bonds that would be between those two amino acids, okay? So you're just building your protein as a group. So I want you to pause the video, go ahead and build your protein. I have some shapes already pre-cut for you. If for some reason you are missing some shapes, because I did do this with my AP biology class as well. So if you're missing some shapes, if there are not enough of a color, there's some scissors in my cabinet. You're welcome to the scissors, and I will leave out some extra construction paper, and you can cut your own shapes. Just go by the instructions and what shape represents each amino acid. Okay, the last part of this is trying to figure out what kind of mutation that you have on your protein. All of you have some sort of mutation on your particular protein. Okay, it is either going to be a point mutation, and remember, a point mutation is where one nucleotide is incorrect, okay? Or it can be what is called a reading frame shift mutation. And that is where you get something that's incorrect and then it messes up every single amino acid after that. The whole rest of the thing is messed up. Okay, and there are then some specific examples of point mutations and frame shift mutations in your notes, and we will talk about those tomorrow. So I have left my original strand with Miss Sanders, and my strand that I have made is what the correct DNA sequence would have looked like in the form of a protein, okay? So mine is the correct protein that was supposed to be made. But remember, you all have a mutation. So I want you to compare your protein that you made to my protein that was made and figure out where your DNA strand went wrong, where your messenger RNA went wrong, where your amino acids went wrong. Okay, so you can compare your codons to mine, compare your shapes and amino acids to mine. Try to figure out exactly what went wrong. And all I want you to do today is determine if it was a point mutation or a frame shift mutation. Is single thing okay except that you have one letter wrong in a codon, which would be a point? Or did something go wrong and it messed up the entire rest of the amino acid? Was there a letter inserted? Was there a letter deleted? And then everything after that got messed up, okay? So then I want you to write that on your instruction sheet. Put on there either where on the side where you wrote your name and your heading, I want you to tell me point mutation or reading frame shift mutation.